Welcome everyone, my name is Michał, I'm Ensembl Outreach Officer, and today we'll be talking about Ensembl's BioMart. So our online export tool. So far we've been focusing on different data types that you can find on Ensembl, but today we'll be talking about how you can retrieve that data on a larger scale. So what is BioMart? BioMart is our online export tool which allows export of large amount of Ensembl data in one go with no programming skills required whatsoever. All you have to do is to follow this kind of point and click web interface to build your queries with a couple of mouse clicks. In other words, it's also a custom download which allows you to download custom tables and sequence files. BioMart's name has been derived from this shopping analogy, which is quite a nice metaphor. So you could be shopping by visiting those small corner shops, specializing in one type of objects, like for example, chemists or pharmacies or butchers, and so on and so forth. Just like you could be hopping from one Ensembl tab to another, collecting or downloading individual objects. Alternatively, you could be using BioMart, which is more of a supermarket where you can retrieve multiple different objects of all sorts in one go. So why would you use BioMart? Why is it useful? Why is it helpful? It's very handy for things that will be really time-consuming or tedious, difficult with the Ensembl browser alone. So imagine you wanted to retrieve hundreds of gene sequences or genomic locations of hundreds of genetic variants. And you could do that using the Ensembl browser by searching for individual genes and downloading the data you wanted and doing it one by one. But that would take you ages. While in BioMart, you could do that in one go. It's also great when it comes to ID conversion. So for example, as the researchers, you might end up working with different resources. All of them have different identification systems and different IDs, and then you end up mapping between different IDs, and BioMart can do that for you at a larger scale. However, BioMart has its own limits as well. It's not designed as whole genome download tool. If you wanted whole genome sequence, please go to our FTP site. When it comes to BioMart search, we recommend around 500 entries at a time. You can find BioMart in the navigation bar at the top of the page in both Ensembl and Ensembl genomes. This bar is blue in Ensembl and it's colored depending on the division in Ensembl genomes. BioMart is available for all vertebrates in Ensembl and most of the non-vertebrates in Ensembl genomes. Except bacteria, it's simply just too many of them. However, it might be possible in our new website, Ensembl 2020. Let's talk about using BioMart now. To use BioMart, you have to follow 4 easy steps. And here we have the overview. You first have to choose the dataset and database you want to search. You then have to provide your filters, so your input that you want to search with. You then have to specify your attributes, or in other words, your output, things you want to print in your tables. And finally, you just have to view and export your results. And let's talk about those steps in more details now. So you first have to specify the database you want to search with your filters. And you can choose from a few databases. You can select the database of all Ensembl genes or genetic variants, regulatory features, or mouse strains. You would only choose mouse strains if you were interested in specific differences between different mouse strains. For anything else, you're more likely to be using mouse genes or mouse variation databases. After choosing your database, you then have to specify your dataset, so the species of interest. You then have to define your filters, so the things you want to search with. Because you're not interested, let's say, in the database of all human genes. You want to narrow it down with your filters to a subset of genes that you're interested in. Like, for example, you might be interested only in genes falling in specific genetic region, or you might be interested in genes matching certain names or IDs, or associated with certain functions or phenotypes. And you can build simple queries by applying only one filter, but you can also build a bit more complicated queries applying multiple filters, which is great as BioMart offers you quite a lot of flexibility when it comes to choosing your filters. And what is important is that the filters follow AND logic. So what you're getting in your output is the intersection of filters. So for example, let's say if I was interested in genes falling in chromosome 9, that would be my first filter. But also in genes with transmembrane domain, and that would be my second filters, what I'm going to get in my output is all genes on chromosome 9 with transmembrane domain. I hope it makes sense. And sometimes if you're not getting any results, it might be because there's no overlap between your filters. You then have to choose and define your attributes. So again, the data you want to export, you want to print in your table or your output. And there are many attributes you can choose from. We're going to explore them in a minute, but I also encourage you to go and explore on your own, see what's available. There's really quite a few of them, and you can choose from different IDs from many different external databases. You can choose feature attributes, you can choose variant information, you can choose homologs like orthologs and paralogs, but also sequence data. Finally, the last step is to view and export your results. We support different file formats, like for example, for tables, you can choose from HTML, comma-separated files or tab-separated files, but also Excel files. However, bear in mind if you're using Excel and working with genes like, for example, sept or oct, Excel likes to convert them automatically to dates, so watch out. And you can also choose a faster format for sequences. If you're an R user, you might also be interested in BioMart, which is our package for BioMart. We do not develop it, but if you're keen, go check it out. You can find more information under the links below. Finally, if you'd like to know more, go have a look at our publications on BioMart, and please cite them if you're using BioMart. Well, let's move to our demonstration now. We have a short exercise. We are going to look at the set of 6 human genes. We could do it for hundreds of genes. However, for the purpose of this exercise, we're only going to look at 6 genes. And we are given their gene names. And we want to find out their NCBI gene IDs. So we have an example of ID conversion here. We also want to explore their function via Go terms, so gene ontology terms. And finally, we want to also obtain their cDNA sequences. So first thing to do is to define our database and dataset. So here we want to find information about human genes. So that means we're after genes. So we're going to choose the database of Ensembl genes and a dataset of human genes specifically. Then we are not interested in all possible human genes, right? We want to narrow down this huge dataset to our small set of 6 genes that match those given names. Therefore, gene name is going to be the only filter in that query. We then have to specify our output, also known as attributes. So things we want to export, and in that case, we've got NCBI IDs, Go terms, cDNA sequences, but also it's advisable to include your input in your output so that you can match your gene names with your obtained output. Therefore, we're going to choose gene names as the attribute as well. And finally, we're going to view and export our data. Okay, great. Let me jump out of my presentation now and copy my input gene names. Let's go to the browser now and navigate to the Ensembl Genome browser. You can go to Ensembl by typing www.ensembl.org. Once you're here on our homepage, you can notice link to a BioMart here in this blue bar on top of the page. I'm just going to click on BioMart, which takes me to our first step, choosing the database and the dataset. So let's click on the drop-down list to see our options. So we can choose from Ensembl genes, mouse strains, Ensembl variation, and Ensembl regulation. 100 stands for the newest Ensembl release, which is release 100. So as you know, in this exercise, we want to look for specific human genes. So we want to search a database of human genes. I'm going to choose Ensembl genes then. And now we can choose our data set, so the species of interest. You can see some popular choices on top, like chicken, human, mouse, rat, and zebrafish. I'm going to go for human genes. So we've chosen our database and data set. Step 1 is done. We can move to step 2 now. As you can see, the filter section has now become available. So we can begin to start choosing our filters. I'm going to click on filters in the left-hand side menu. Filters are here grouped into different categories. So we've got region-related filters, gene-related filters, but also phenotype, gene ontology, or multi-species comparisons, protein domains, and variant-related filters as well. I'm not going to explore all of them now. I encourage you to go explore on your own in your free time. Just play with it, see what's available, how you can apply it in your own research. But let's have a look at a couple of them. So let's expand region filters by clicking on the plus. As you can see, we can narrow down the database of all human genes to genes, for example, only on specific chromosome. To do so, you first have to tick the chromosome filter so that it becomes active, and then choose a specific chromosome, for example, chromosome 7. And you can now see that the filter has now been applied to our search and it's been added to the filter list on your left-hand side. However, in this exercise, we don't need chromosome filters. We are only looking for genes of a specific name. So to get rid of a filter, simply untick it and it's going to disappear from the list. You can also limit your search to more specific genomic regions. So apart from choosing a chromosome, you can add coordinates. You can add start and end coordinate, but you can also search using multiple genetic regions provided as a list here in this text box over here. Let's close that for now. Let's explore the gene filters. Here, for example, we can search for genes matching certain IDs. That's the filter we're going to use in a minute, but let's have a look what else we've got here. You can also limit your search to genes mapping to specific microarray probes. You can search for specific gene types, like for example pseudogenes or long non-coding RNAs, all sorts of other RNAs, protein coding genes, and so on and so forth. You can search for genes of a specific source, and you can also filter based on transcript annotation or transcript scoring algorithms like transcript support level, APRIS main, and GenCode basic. Before we apply our filters, let's have a look at the other categories. Here you can search using specific phenotypes, and also you can search using gene ontology terms. Remember, in this exercise, we want to output gene ontology categories, so gene functions and processes our genes are involved in. However, we are not searching with gene ontology categories. It's important to realize what your filters are and your attributes are and differentiate between them, okay? So filters, again, is our input, things we're searching with, and attributes are our output, so things we want to print in our table. Okay, let's go back to our ID filters. I'm just going to quickly close those. So here we have input external references ID filter. I'm going to select this. And as you can see, by default, this filter is set to Ensembl stable gene IDs, starting with ENSG. However, if I click on this drop-down list, you'll see there's many different IDs you could search with. So you just have to find the appropriate ID category that you want to use. And in this case, we're not searching with Ensembl stable IDs, but with gene names. So I'm gonna look for gene names here in this list, which is over here on top, and I'm gonna click on it. We now need to provide our gene names. So I have them copied. I can just simply paste them here. They can be pasted either as a comma separated, tab separated entries, or as a list. Alternatively, you could also upload them as a file using this browse button underneath. As you can see now, the gene names filter has been applied to our query. Step 2 is done. We can move to step 3, choosing our attributes. However, before doing so, I'm going to show you the count function, which is quite useful after applying your filters, which can show you if your filters have been applied correctly. So if I click on this count button, it's going to show me how many genes match my filters. As you can see, out of over 67,000 of all possible human genes, our filters match 6 genes, which makes perfect sense as we provided 6 gene names. Okay, great. Let's move to step 3, attributes. I'm going to click on attributes now. And first thing to realize here is that we have 6 main attribute types, which are indicated here by those different radio buttons. So we have features, attributes, structures, homologs, variant, including germline and somatic, and sequences. What is important, you can only query one category at a time. Okay, so we know that in this exercise we're interested in feature type attributes like gene ontology terms and external IDs or things like that. However, we also want to get cDNA sequences, which we can find under sequence, which already hints that we'll have to build 2 separate queries. But let's start with features. I'm going to expand the gene to see what we have got over here. You can see there are a few attributes already pre-selected for you. So we've got gene stable ID and its version, but also transcript stable ID and its version, which are already active, listed in our attribute lists. There's also many other attributes you might be interested in. And again, I would encourage you to go and explore in your own time what options you can choose from. Let's say I'm not interested in gene stable ID version or transcript version. I'm going to deselect those and I'm going to search for NCBI gene IDs, as that's what we were asked for. So I just expanded the external features. And here we have a bunch of other attributes we could choose from. And you can already begin to notice that we have all sorts of RefSeq IDs, NCBI IDs, and IDs from many other databases like OMIM. Here we have our NCBI gene ID that we're interested in, so I'm going to select this. Furthermore, we also have gene ontology categories here on top. I'm going to select all gene ontology term accession, name, and definition. The last thing to do is to add our gene names, so our original input, to our output list. It's generally a good practice to add your input to your output so that you can easily link your gene names to your printed output. You can find gene names under the gene attributes. So let's go up and select gene name. Okay, great. So that's step 3, choosing attributes, done. Our attributes list on the left is complete now. We can move to step 4, viewing our results. To view our results, simply click on the result button in the top left. And here we have our results table. So first thing to point out is that you can see our list of attributes matches the columns in this table. So we've got gene stable ID, transcript stable ID, NCBI gene ID, and also gene ontology, terms, accession, name, and definition. So I can scroll down and you can see by default we only show you 10 rows. You can change it. You can choose to display all rows. If you click it, it's going to open a pop-up window displaying all of your results. Here we are. You can see this table is pretty large. Actually, it's quite massive. So why is it that we only inputted 6 gene names, but we have so many rows in our output? It's because each of those genes might have multiple transcripts, and every transcript can have multiple associated gene ontology terms. That's why this table ends up being quite big. Okay, let's go back to our original table, as there's one other thing I wanted to show you. So you're probably interested in having this data in a file on your machine to work with it rather than having it online. To do so, you have to export the data. You can do it using this export all results to a file option. You can choose different file formats like tab separated, comma separated files, HTML or Excel files. Furthermore, you can also choose to export compressed file or to export compressed web file notified by email. It's a really nice option if your query is quite big. What it does, it prepares the file and packages it at our end, and then when it's ready, we send it to you via email, which is quite nice as you don't have to do it locally on your own machine, because when connection drops, you might get an error or truncated file. All you have to do is just to provide your email in this box below and then hit go. I'm not going to do it now as we have a second query. We want to get our cDNA sequences. So there's no need to start from scratch. You could, if you wanted to, you can hit on new. However, we can go back straight to attributes by clicking on the attribute section. Here I'm going to change the attribute types from features to sequences. And let's now explore our sequences option. As you can see, there are different types of sequence you can export. By default, we have peptide sequence pre-selected, but you can change it to any other type. And when you do so, you can see the diagram changes to visualize the type of the sequence. We want to get cDNA sequence, so I'm going to select that. And then we can also modify header information. So let's expand the header and see what we've got here. So as before, we've got gene stable ID and a version pre-selected, as well as transcript stable ID and its version. I'm going to deselect those as I'm not particularly interested in them. But then it's also nice, again, to include your input in your output so that we can match up our gene names with the outputted sequences. So I'm going to select gene name as well. As you can see, our attributes have been added to the list, so we are ready to click result. Just as before, we only show you first 10 entries, and you can see they're quite long. And again, you can see them all by changing the view option. If I click on all, you'll see that again there is quite a few sequences provided here. And again, why so many sequences while we only have 6 genes? Is because each of them can have multiple transcripts, and all the transcript sequences are listed here. And finally, you can also export your sequences as the FASTA file. Okay, great, that's all I wanted to show you today. I hope you found it useful and that you can start applying it in your own research.